Welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. This is lecture number 57, where under the broad domain of phenolics, I will now discuss about the biosynthesis of volatile benzenoids. In the previous class, we talked about volatile phenolics. So, here mostly focus will be on the benzenoids, that is C6C1 metabolites. So, the concepts to be covered: the origin of benzenoids from phenylpropanoid, volatile benzenoids, that is floral scent, the plant species Clarkia and Petunia, these were used as a model plant, and what has been discovered with this plant I will try to present. Then after that I will talk about the benzenoid network in plants, and then once we discuss all these things, then I will discuss little bit of the work what my lab is doing on the benzenoid volatiles with a very popular and Indian Indian fragrant flower, which is Jasminum sambac or belly or bella. Okay. And then we will also talk about the emission mechanism of this benzenoid volatile emission, particularly in terms of physiology. So, hope you will find it interesting as well. So, again the point is that the simple phenolics C6C3. Okay. So, this is benzenoid means it will become C6C1. So, that will be our point of discussion. So, this is basically one paper which published. Okay, there is a mistake here, I must correct it. So, this is a paper published in plant physiology, not in plant cell environment. I think this is volume numbers are fine, only the name of the journal. Anyway, now I have corrected it. So, what we see here is basically the transenamic acid starting point of the phenylpropanoid pathway, and which makes paracoumaric acid by cinnamate 4 hydroxylase, which should be here. And then mind it, this is the year of 2000. So, I have picked up intentionally this paper. So, where it was proposed that from paracoumaric acid eugenol is formed by multiple step reaction. At that point of time it was not clear so how it is formed, but now we know that basically the ferrural CoA, basically the ferrural CoA contributes towards the formation of monolignol pathway leading to coniferyl alcohol, and that makes eugenol, whereas paracoumaryl alcohol also contributes to chavicol. So, the point is this: at that time point the eugenol to methyl eugenol, that is called isoeugenol methyl transferase, that has been characterized, and BEAT, as I said, that is the first enzyme discovered. Later the BHD name was given, but BEAT was discovered in this context with the plant species called Clarkia breweri, Onagraceae family. It produces a big flower which emit both benzenoid and terpenoid volatiles. So, when they analyzed the benzenoid volatiles, they found that that was dominated by benzyl acetate, along with that methyl eugenol, methyl salicylate, methyl benzoate. So, these are the components which were dominated in the scent bouquet of Clarkia breweri flower. So, once they have identified this compound by GCMS analysis, they try to work out how this is formed, and accordingly what was clear at this was the methyl. So, there must be a methylation reaction, similar like methyl isoeugenol was detected, which must have produced from isoeugenol. So, isoeugenol methyl transferase will be there. Similarly, eugenol will convert to methyl eugenol by a similar enzyme, and benzyl acetate, in the context of benzyl acetate, the BEAT is a totally a novel enzyme was discovered, which converts benzyl alcohol to benzyl acetate, and we know that in this conversion it requires a acetyl CoA. Similarly, Methyl salicylate was also discovered as one of the volatile components. So, in this context salicylic acid methyl transferase or it is later named as BSMT, benzyl salicylate methyl transferase has been characterized, and methyl benzoate was also found. So, the benzoic acid methyl transferase was also subsequently characterized. So, I mean here that BAMT, SAMT, BEAT, IEMT, all these enzymes and subsequently genes were characterized from Clarkia brewery flowers. And now, what we see here is this, that the salicylic acid carboxyl methyl transferase, which I said SAMT, the name is like this. So, this what you see that the one is the day 1, that is the petal time post anthesis day 1, that means the when the flower bud opens fully bloom. So, and -1 is the 1 day before, -2 is 2 days before. So, what you see that the maximum activity was found just 1 day before, particularly for the SAMT activity, and then once it opens the activity declines, okay, and then it further declines. Whereas, but in case of beet it is a different trend. So, the flower opens at 1, that is 1 day time post anthesis, and it continuously enhances its activity, leading to the formation of benzyl acetate. So, that means that as long as the flower alives for 3-4 days, it continuously emit benzyl acetate, and there is maybe a relationship between benzyl acetate and pollinator attraction, that is another aspect. And for but in case of isoeugenol methyl transferase, you see the maximum activity was found here at day 1. So, that means this enzyme the physiology of this enzyme activity differs from enzyme to enzyme, and that means how the composition differs with time. At certain point of time it may be more, just at the time of opening it may be more of your methyl eugenol or isomethyl eugenol, and then that level more or less maintained. But what happens, but the SAMT which was maximum just before the day of opening, with time it releases down. So, there are physiological significance, but that is not our point of discussion. Next, what we see, this one discovery in 2006, where they have characterized just a year before they characterized BPBT or BEBT. I have mentioned in the previous class that this requires benzoyl CoA and benzyl alcohol, which joins this again a BAHD family enzyme, which is BPBT, and it makes benzyl benzoate. Similarly, benzoyl CoA can also joins with phenyl ethanol, that is also an alcohol, and makes phenyl ethyl benzoate. So, in 2005 this enzyme was characterized at the molecular level, and what we see in 2006, what they have done, they try to block this activity using RNAi approach, okay, and they want to see what is going to happen as a result of this blockage. And what has been found, that when elimination of benzyl benzoate formation decrease the endogenous pool of benzoic acid and methyl benzoate. So, elimination of benzyl benzoate means that the benzoyl benzoate content is getting down. Why? Because BPBT is blocked. When BPBT is blocked, then what will happen? It decrease the endogenous pool of benzoic acid and methyl benzoate. So, that is interesting, that these two contents are also getting decreased. Okay. So, earlier what has been thought that benzoic acid is formed through this pathway, benzaldehyde to benzoic acid, benzaldehyde is formed from cinnamic acid, okay, and then benzoic acid is formed, it converted into methyl benzoate. But now, with blocking of BPBT, what has been found, both BA and methyl benzoate has decreased. So, that means there must be an alternative route for the formation of benzoic acid. That means, there must be, and let me erase this, there must be an alternative route for the formation of benzoic acid from benzyl benzoate. So, which is yet to be characterized. So, this confirms that contribution of benzyl benzoate towards benzoic acid formation, that is entirely a new hypothesis which is coming out from this work, and that is why. So, next point is this, look, what I have said even in the previous one, that all is started with phenylalanine. Although phenylalanine I have not shown here, it is not shown here, but you can add here PHE and put a arrow and put the enzyme PAL. So, phenylalanine, and we know how phenylalanine is formed. Phenylalanine is formed from cyclimate arogenate pathway. Okay. So, now, cyclimate arogenate pathway. So, phenylalanine, here is your phenylalanine and here is the tyrosine. It comes from arogenate, and arogenate is basically coming from prephenate, and prephenate is coming from chorismate. Okay. So, this is basically a paper which published in 2010 by the same group, where what they have done. So, they characterize a arogenate dehydratase. So, arogenate dehydratase is here ADT. So, they block ADT, and by blocking this, RNAi suppression reveals phenylalanine is synthesized by the arogenate pathway. Okay. So, let me read the statement that ADT1 suppression in petunia petal significantly reduce the ADT activity, which is clear, and levels of phenylalanine, of course, phenylalanine level will be reduced, and the downstream phenylpropanoate benzenoid volatiles. That means, the benzenoid volatiles also will be reduced, and but unexpectedly arogenate levels were unaltered, while cyclimate and tryptophan level were decreased. So, tryptophan level decreased, cyclimic acid level decreased, but arogenate level remained unaltered, which is difficult to explain, but feeding with cyclimate led to the formation of prephenate and phenyl pyruvate accumulation. That means, when cyclimic acid feeding was done, so that leads to formation of both the things, both phenyl pyruvate as well as prephenate, and that leads to partial recovery of phenylalanine, which suggests that this may be another alternative route of phenylalanine formation. Although the main route of phenylalanine formation is this route, that is from cyclimate, chorismate, prephenate, arogenate phenylalanine, but there may be another route involving from prephenate to phenyl pyruvate can contribute to the phenylalanine. And another important point is this, the regulation lies upstream of phenylalanine. That means, the volatile emission in petunia is basically regulation lies upstream of phenylalanine, that is also very interesting. So, it is far ahead of this. So, another we see another interesting work here in that published in Nature Chemical Biology in the next year, where they have characterized prephenate aminotransferase. So, prephenate aminotransferase is basically it was not characterized before, and that was basically upon this characterization where scientists claim that they have now completely understand the pathway of phenylalanine formation from the DHP starting with the DHP synthase. So, prephenate aminotransferase directs the carbon flux from prephenate towards arogenate and which is predominated in the phenylalanine biosynthesis. So, that is interesting, and what they have done, they try to block these activities, and as a result of this they found that it substantially reduced the benzenoid volatiles levels in the petunia flower. So, we started talking about benzenoids, benzyl benzoate. There are two benzene ring, but it's all started with C6C1. So, this is benzyl benzoate and this is phenyl ethyl benzoate. So, you can also write PEB, it's fine. So, and the BPPT enzyme is basically either joining phenyl ethanol with benzyl CoA or it joins benzyl CoA with benzyl alcohol and it is making benzyl benzoate. Okay. So, this is well characterized and also BSMT is well characterized and benzaldehyde to benzoic acid, which is simply a aldehyde dehydrogenase, which has also been characterized. Okay. And cinnamate to cinnamyl CoA, which was also characterized or sorry, this is kumaric acid to kumaryl CoA, which was by a 4-Cl enzyme, which is well characterized, and this pathway eventually leads to isoeugenol or eugenol that we have seen in the previous class. So, that is fine, but the point here the networking says that from this kumaric acid to the benzaldehyde formation, that means kumaric acid is C6C3, this is C6C3 to when it comes to benzaldehyde which is C6C1, all this C6C1. So, this is a chain shortening. So, there may be 3 different ways these things can happen. Okay. One is called non-beta oxidative route or non-oxidative route, where there is no involvement of as such the CoA thioester. Okay. So, where straight away kumaric acid can form benzaldehyde by through the formation of one unstable intermediate, which is 3-hydroxy 3-phenyl propionic acid, this one intermediate, and it makes benzaldehyde, and benzaldehyde subsequently converted to benzoic acid subsequently converted into methyl benzoate. And what we have learned in the previous slide, that benzyl benzoate also contributes to benzyl alcohol benzoic acid, but this has not yet been fully characterized. Now, the other point is that one is this is non-oxidative, and now the other one is the core beta oxidative pathway. So, here scientists put lot of effort and finally, they made this pathway confirmed. So, first enzyme what they have characterized, which was called CNL, which converts this cinnamic acid, this is cinnamic acid to, okay, I made a slight mistake. So, this is basically cinnamyl CoA, this is not kumaryl, because cinnamyl CoA straight away forms to benzaldehyde, not kumaryl. So, kumaryl CoA will form the 4-hydroxybenzaldehyde. So, this is cinnamyl. So, this is cinnamyl CoA, this is cinnamic acid, this is cinnamyl CoA. So, cinnamyl CoA will enter into the peroxisome, and then there is an enzyme called CNL, which is similar to 4-Cl, which is cinnamyl CoA ligase, which converts cinnamic acid to cinnamyl CoA, and then this cinnamyl CoA subsequently forms through a beta oxidative pathway benzoyl CoA. So, that is 3-hydroxy 3-phenyl propanol CoA, and then 3-O means 3-oxo 3-phenyl propanol CoA, and then it makes benzoyl CoA. And what has been found with time, that this is a very unstable compound. So, cinnamyl CoA can form to 3-oxy 3-cinnamyl 3-oxy phenyl propanyl CoA as a single step, and in the next slide we will see what is the enzyme. But at that point of time when they have published this paper in 2012, so they have characterized this KAT, which is ketothiolase. So, ketothiolase is basically doing the reaction which converts 3-oxo 3-phenyl propanyl CoA into benzoyl CoA. So, this has been characterized in this work. So, and this is what happens. Let us look inside the peroxisome. So, cinnamic acid enters through some transporter which is not clear, then core beta oxidative pathway is operating which makes benzoyl CoA. So, the C6 C3 now becomes C6 C1. Okay, then from benzoyl CoA it makes benzoic acid. T stands for thioesterase. So, the CoA will be broken down. So, benzoic acid is formed. This benzoic acid may come out into the cytosol and converted into benzoyl CoA. So, here is a BZL, this is benzoyl CoA ligase, or there may be a CoA ligase exist here which straight away convert benzoyl CoA to benzoic acid to benzoyl CoA without the involvement any transporter. So, this is the possible route of benzoic acid trafficking. The only point is this, the benzoic acid is a very simple molecule. Okay. So, this is benzoic acid, but you see that its formation is so complex. Okay. Here in the past, okay, in 2009, so the ketothiolase has been characterized as I have mentioned, which converts 3-oxo 3-phenyl propanyl CoA, which is a C6 C3 compound, into benzoyl CoA, which is C6 C1. So, here the chain shortening occurs. So, this is C3 chain is now becoming C1 chain. So, this was well characterized and that was published in PNAS in 2009. Okay. So, next one is BPBT we have discussed, we will not discuss. Now, come to more or less this one. So, where they mentioned here is that they confirm the presence of a cinnamyl CoA ligase which converts cinnamic acid into cinnamyl CoA, but this cinnamyl CoA ligase is basically stays inside the peroxisome. Okay. And but also it is there is a possibility that a non-oxidative route is also functioning, as this cinnamic acid converted into via non-beta oxidative route to benzaldehyde, which I have mentioned, and this benzaldehyde to benzoic acid, then methyl benzoate is all formed. So, here basically they are confirming that CNL or the cinnamyl CoA ligase plays an important role leading to the formation of benzoyl CoA, and all 4 CLs are very important, CoA ligases are very important. So, basically they play with that and see that how blocking 4 CL really affect the pathway. And then finally, why this is in plant cell environment published in 2014. Now, this is the clear picture. So, what we see here is cinnamic acid is in the cytosol that moves inside, and then CNL enzyme is there which converts the cinnamic acid into cinnamyl CoA, and then there is a CHD. So, this is cinnamyl CoA hydratase dehydrogenase. So, which converts cinnamyl CoA into 3-oxo 3-phenyl propanyl CoA. So, in between there is an intermediate, I say 3-hydroxy 3-phenyl propanyl CoA, but that is an intermediate. So, this basically enzyme catalyzes these 2 reactions, but for the simplicity cinnamyl CoA, if you incubate cinnamyl CoA with this enzyme, you will straight away find 3O3PP CoA, and this subsequently by a ketothiolase make benzoyl CoA. So, here is the chain shortening occurs, this is the chain shortening enzyme here, and then this benzoyl CoA will move out and utilized for formation of phenyl ethyl benzoate or benzyl benzoate as the case, or it may also leaving the CoA out can form the benzoic acid, and benzoic acid can subsequently utilize for methyl benzoate and so on. Similarly, the cinnamic acid can convert it into paracoumaric acid, and paracoumaric acid to this ultimately it forms coniferyl alcohol or it and through multiple route. So, which is the monolignol route. So, that we are not discussing here, but then CFAT I have discussed in the last class, coniferyl alcohol acetyl transferase and then which makes coniferyl acetate. This is coniferyl acetate, and then their eugenol synthase will make eugenol, isoeugenol synthase will make isoeugenol, and then a subsequent methyl transferase will make either methyl eugenol or methyl isomethyl eugenol as the case may be. So, this is in brief the latest update of the benzoic acid biosynthesis. Again, as I said, the benzoic acid is a very small molecule, but this benzoic acid formation requires such a complex route. Like you remember the monolignol pathway. So, when we introduced the meta hydroxylation, so formation of caffeate from coumarate simply it requires a 3 position OH, but it is not that easy. It involves joining with either coumarate or joining with either quinate or sikimate, and then there will be cleavage and then there will be hydroxylation, it forms the subsequent caffeoyl moiety and then subsequent conversion. So, that means apparently the structure looks very simple. So, chemically it is possible to make this to this, but in the biological system it is pretty complicated. So, with this I now move into the work what we have done in past years regarding the scent volatiles emitted by Jasminum species. So, Jasminum species like I mentioned belly or bella, then there is another called juthi or jui. So, the belly is Jasminium sambac and the jui is the Jasminium auriculatum. So, this is a holistic schematic diagram what is showing here that the volatile scent volatile consists of both terpenoids and benzenoids. So, in this class we are focusing on the benzenoids because terpenoid aspect I have covered in previous classes. So, what is showing here that multiple cellular organisms play an important role in the synthesis and storage of the volatiles. So, it is stored sometimes as glycosylated volatiles, that means it joins with a glucose molecule, and as and when required there will be deglycosylation which is by the glycosidase enzymes, and as a result of that this will be emitted and the emitted volatiles will subsequently released. Okay. So, this is a general. So, now I say that when we work with Jasminum sambac, so Jasminum sambac actually it remains alive for more than 24 hours. So, we saw how the flower changes starting from the bud to the full opening until senescence, how it changes, how it openings the petals, and its petals have multiple holes, 2 or 3 layers of petals, and how the changes occur throughout the floral lifespan is basically showing here. And then what we have seen here, look, the benzyl acetate, now you are aware about benzyl acetate is found in Clarkia brewery. So, from there the BEAT has been characterized, and also in Petunia. So, now here also in Jasminum sambac we also detected a huge amount of benzyl acetate, and basically then we detected in the flower how the benzyl acetate content changes with time, and this shaded portion are is the night time, because it is a white flower it emits benzenoids. So, benzenoids are usually emitted at night time which attracts the moth. So, all these things that maximum emission occurs in the night time, and that is dominated by the benzyl acetate, then methyl benzoate, then methyl salicylate. So, this is the typical flower of Jasminum sambac. So, now not all benzyl acetate is emitted. So, or even benzyl, if benzyl acetate is emitted, but benzyl alcohol it prefer to stay as internal pool. So, it is not emitting, but it is staying there. So, we also measured that what is the content of this internal pool of volatile compounds throughout the floral lifespan. So, it is like this. So, a substantial amount of benzyl acetate also accumulates as internal pool, later it is emitted out. So, this benzyl acetate may store as unconjugated volatile and later it emitted, or it may undergo glycosylation also. Okay. So, this is another simplified photographs of Jasminum samba flower stage wise. So, S1, S2, S3, S4, S5, S6. So, that is that stage 1 is at 2 p.m. day 1 when the bud is still growing, and then H2 is basically the 8 p.m. and then S3 is basically 2 a.m. at the midnight when it is about to open, and then S4 stage at 4 a.m. 8 a.m. in the morning when it is opening, and then S5 is fully opened, and then S6 that is now it is ready to undergo senescence. So, at these 6 different stages we studied. So, this is one more thing that the, as I said, benzyl alcohol it undergo glycosylation. So, a glucose moiety is added and then it is stored in the floral tissue, particularly in the vacuole, and then upon requirement deglycosylation occur by enzymatic reaction, leading to the emission of free endogenous volatiles. And at that 6 stages, that means the stages are here S5 and S6. Okay. So, the phenylalanine ammonia lyase, because that is the PAL enzyme which is the starting point for these benzenoids. So, where it remains maximum? It remains maximum just at the time of opening of the flower at S3 stage. Okay. And whereas the beat activity detectable beat activity is there, although maximum beat activity occurred at the S4 stage, that means at this stage maximum beat activity, but maximum PAL activity is here. Okay. PAL activity first is required because PAL will come first, it makes cinnamic acid, and then later the beat will make the subsequent benzyl acetate. So, that makes sense. So, more PAL activity is the earlier stage and beat activity will be the later stage, and this is basically the glucosidase activity that is more or less constant throughout the stages of floral lifespan. Now, this is interesting, you see that I will only show the PAL. So, this is night. So, PAL activity reaches its maximum here at 12 o'clock midnight or so, and then it's continuously coming down again in the next day evening, it's slightly reaching up. So, that is important because PAL activity and then followed by the other reaction leading to the formation of the phenylpropanoid pathway, and this is the beat activity, but it reaches maximum much later than that of the PAL activity. And then benzoic acid methyl transferase, which converts benzoic acid to methyl benzoate, that has also been measured throughout the floral lifespan, and here you see the maximum activity during the night time. This is the night time, whereas salicylic acid methyl transferase maximum activity occurred in the daytime, and that is but during the bud condition. So, I have shown that salicylic acid methyl transferase is more during the bud time. So, if you see in case of the previous slide, if I remember correctly, here also maximum salicylic acid methyl transferase occurred at -1, that is before the opening. A similar thing also we observed in our case as well. This is the time petal opening and this is before. So, and this is basically a nutshell about the major volatile compounds emitted by the Jasminum sambac flower, and also we have characterized partially that a transcription factor like odorant plays important role. So, odorant expression basically correlates with the expression of benzenoids. So, we have partially characterized the odorant transcription factor, and finally, based on the different study like volatile emission, enzyme activity, like gene expression, then a holistic mechanism was presented here, what happens in the cell. So, that the volatile compounds are synthesized in the cell, they stored in the vacuole, later they release the free volatiles internal pool, okay, and then they released as the emitted volatiles. So, there is a role of transporters, because we also characterize a few transporter genes. We have studied the expression based on the transcriptomic information available. So, and so this is basically in a nutshell what happens in the Jasminum sambac flower, leading to the emission of volatile and volatile, which dominated by benzenoids, but also it also constitutes terpenoid component as well, dominated by linalool. So, this is in general the outline of mechanism governing Sen volatile synthesis, storage and emission in Jasminum sambac. So, do not get daunted, it looks pretty heavy, but this is just to give you an idea about how reaction happens. So, and that leads to scent. So, the scent means what you get it in your olfactory nerve through nose. So, it is not a single component. So, it may be starting from 20 to 60, 70 components, that all constitute that specific scent. So, that is the message. So, with this I end this class. Thank you.